J'attendais qu'ils viennent de là.
Je vais garder le mien parce que j'étais en contact. Je suis négatif. Hein, mais
Hello everyone. Let's start the session. We have a packed schedule today. So my name is Paule-J Toussaint. I work at McGill University in Montreal. We are chairing the session Neuroanatomy and Physiology. So if you did not want to see this one, please leave now. Our first speaker is Justine Hansen from McGill University, who will present a multi-institutional international initiative aimed at mapping neurotransmitter systems to the structural and functional organization of the human brain, of human cortex, sorry. Justine.
Hello. Okay, yes, you already have my title, so I'm going to just skip that. So this is a very scientifically accurate diagram trying to illustrate how a receptor might respond to a neurotransmitter, the binding of a neurotransmitter, which is going to change how ions are flowing in and out of the cell, and ultimately whether this neuron is going to fire an action potential or not. So that makes that makes these receptors really key modulators of firing rate, and by extension, if we spread our imagination a little bit, also of brain connectivity, so whether a neuron is going to synapse onto another neuron, and of dynamics. So I wanted to take this idea outside of this single cell level and try to explore whether I could find this at the level of the whole cortex. So do I find that the distribution of different neurotransmitter receptor densities, is that going to affect how the cortex is connected and the function? So to do that, I needed data. And I'm not a PET expert, so I did not collect this. I sent emails to PET researchers and asked them if they would be willing to share their group average PET tracer data with me, mapping receptor distributions, and they were extremely generous. So the lesson there is that researchers are actually much more generous than you think. We ended up collecting PET tracers for 19 neurotransmitter receptors and transporters across 9 neurotransmitter systems, a whole bunch of healthy controls. This data is openly available. I got permission to share them, so it's at my GitHub, but also on the Python software toolbox NeuroMaps. When I was doing these analyses, comparing this PET atlas of receptor distributions to brain structure and function, I had this replication data set coming from Carl Willis and Nicola Palomero-Gallagher, she's sitting right there, from autoradiography binding. So the first thing that I did with this was I find that the receptor gradient separates classes of laminar differentiation and is associated with synapse density. So what do I mean by that? Here is this receptor gradient. So this is the first principal component of receptor density. And you can see it visually, but also when I kind of separate it across the different classes of laminar differentiation, so how the cells are distributed in the layers of the cortex. And it separates idiotypic from paralimbic regions, and is also correlated with the synapse density, which is kind of cool considering previous work that shows a relationship between spine count and the receptor gradient in macaques. The second thing I do is I look at this thing called receptor similarity. So I'm going to explain that a bit. You can imagine at every brain region there's this like fingerprint of receptors. So I had 19 receptors in my atlas, so there's a density for each one, and if I pick 2 brain regions and correlate them, I'm going to end up with this measure of how similar 2 brain regions are in terms of their receptor fingerprint. And then I do that for all the brain regions, and I get this region by region receptor similarity matrix. So quantitatively, this is just a correlation between how similar the fingerprints are, but conceptually, this is like how similar my 2 brain regions respond to the same neural signal. How similar are their ear profiles? Like, what are they listening to? Using this receptor similarity matrix, I find that neurotransmitter systems are systematically aligned with structural and functional connectivity. So I'm comparing this to structural connectivity. Did you see it? Yes, okay. Structural connectivity, functional connectivity, and I'm asking questions like, if 2 brain regions are connected, do they have more similar receptor densities? And if 2 brain regions are in the same, sorry, this clicker is not working there. If 2 brain regions are in the same functional network, do they have more similar receptor profiles? I also find that the receptor similarity is correlated with structural connectivity and with functional connectivity after regressing distance out of these. So this is above and beyond this common phenomenon that we find in the brain, that brain regions that are close together tend to have similar profiles. The next thing I looked at, so I kind of situated these receptor densities within structure and function, but I also kind of intuitively knew that these receptors have a lot to do with the firing rate of the cell, and so do we also find this at the macro scale level? Are we finding that these receptor densities have something to do with oscillations? So here what I'm doing is I'm using MEG power distribution bands from HTP. So every single row is a different power band and also a different multilinear regression model where the input variables are the receptors. And the output variable is that power distribution band. Each one has a fit or an adjusted R squared. And first of all, the fits are quite nice and they cross-validate nicely. So that was cool to see that there's this robust relationship between the densities of these receptors and then oscillatory dynamics. And then I applied this thing called dominance analysis, which distributes the R squared, or the fit, across the input variables. So it gives me an intuitive idea of what input variables are contributing more or less to this model. And I see some things pop out like the mu opioid receptor, but I also am seeing ionotropic receptors like alpha-4 beta-2 and GABA show up. And that result is even stronger in the autoradiography data set simply because I have more ionotropic receptors there, and I'm finding things like AMPA and MDA kainate, et cetera. That was kind of nice because I know that these ionotropic receptors are those that respond faster on a shorter time scale to the binding of this neurotransmitter. So it's cool that there's this link between the ionotropic receptors and these fast oscillatory dynamics coming from MEG. So I'm going to take a little step back now and talk about the relationship between these receptor densities and microarray gene expression. The reason I did this was that there was work being done that was using gene expression from the Allen Human Brain Atlas as a proxy for these receptor densities, which is totally fair game considering it was difficult to get your hands on the receptor densities themselves. So I just wanted to know, is that assumption valid? And I actually find that in many cases there is a poor spatial correspondence, just a poor correlation between the two. That's all the blue scatter plots. So those are those where the protein density and the gene expression coming from microarray gene expression are not very correlated. And I tried to dig a little bit deeper into why that might be the case. And I find that it could be to do with intersubject variability. So in the next plot, I am on the x-axis plotting the correlation between the gene expression and the receptor density. And on the y-axis, that is the differential stability of the gene that is coding for the receptor. So that's a measure of how variable this gene is across the 6 donor brains in the Allen Human Brain Atlas. So something like 5-HT1A, that's a serotonin receptor. This one has a great correlation with its gene expression. So the PET map, the receptor density map, is nicely correlated with the gene expression. And also, this gene expression map is quite consistent across the donors. So suggesting this relationship between intersubject variability and how closely the two correspond to one another. So the last thing I'm going to show you is my kind of cherry on top. I had looked at structure and function. I have looked at dynamics. I've looked at gene expression. Considering how the receptors are so well situated within this microscale architecture of the brain, the next kind of logical step was, well, it must be supporting brain function somehow. It must be supporting cognition somehow. So to try to make this link, I was using a partial least squares analysis between the receptor densities on one hand and neurosynth meta-analytic functional activations on the other hand. So that's a mapping between a brain region and a cognitive term, a cognitive process. I get one significant latent variable, and this represents a relationship. Sorry, I've clicked this many times. There we go. This represents a topographic gradient across the brain that is linking these receptor densities with these cognitive patterns. And this model is cross-validated using a distance-dependent method. And then the most interesting part is which receptors are contributing the most to that gradient and which cognitive processes are contributing to that gradient. So in other words, what receptors are covariing with what functional activation patterns associated with certain cognitive processes to give me this nice gradient. And I'm finding that on the receptor side, I'm looking at especially serotonergic and dopaminergic receptors. And on the cognitive loading side, I'm looking at things to do with affective psychological processing. So empathy, stress, anxiety, emotion, et cetera. So this is a nice bridge between these monoamine receptors, these terms to do with affect, and they spatially covary with one another according to this gradient. So that brings me to the end. We collect a bunch of PET receptor maps, and we do make them available, so feel free to use them. We situate chemoarchitecture within the multiscale organization of the brain. Neurotransmitter receptor profiles closely align with the structural connectivity of the brain and mediate its link with function. And lastly, the overlapping topographic distributions of these receptors ultimately manifest as patterns of cognitive specialization. Code and data are on GitHub. Also on that Python software toolbox I mentioned before, Neuromaps, which does have an associated poster, so you can't see me there. I have more than one poster, so if you don't see me at one, I'll probably at the other one. And yeah, thank you very much for listening to my lab. This work is a preprint. The gene expression work is actually its own preprint. Yeah, that's all. Thank you.
We have the time for one question. So if you Want to please write your question in the app? Otherwise, I will ask a question. There we go. We got one. Do you think that lack of correlation between PET and Allen Institute are due to differences in protein versus mRNA expression?
Yeah, I think there can be many reasons. I think it could be a methodological thing, but I definitely think there are very many reasons that the gene transcript is not necessarily correlated to the protein density. So I think that assumption in and of itself is not necessarily, obvious. For example, protein turnover, regulatory mechanisms, there are for sure reasons that the gene transcript might not correspond to how much protein is actually being expressed at the cell surface.
Very good. And well, I have another question. Did you look at the pattern of receptors that you have and compare it with other maps, such as the one in Jülich that are like post-mortem?
Sorry, I didn't understand.
Did you compare your pattern of receptors with the one from Jülich?
Oh,
they have post-mortem maps, and does it match? Or
Yeah. So the PET tracers, many of them match well. So for example, you have a mu-opioid receptor, that one looks great. 5-HT1A looks the same wherever you look at it. There's a couple less so. So for example, 5-HT1B, I actually have, in that data set that I present at my GitHub, it's more than just what I showed because I had some cases where I had multiple tracers and I had to pick one. I didn't want to combine them. And for example, 5-HT1B, that one's a little trickier. Sometimes it doesn't correspond as nicely, so I had to make some decisions about which one to choose. But yeah, so the answer is for some, yes. For others, less so. Yeah.
Thank you very much.
Thank you.
So, apologies everyone, I forgot to present my two co-chairs, the two to my right. You have our future chair, Michel Thiebaud de Chetaine, and to his right, you have, Nicolas Palomero Gallagher, who you must have seen her name come up in your ballot recently, so you must have voted. Hint, hint. So, our second speaker is Karen Saltoun, also from McGill University, who will speak about a second look at the second half, uncovering brain global asymmetry patterns at the population level.
All right. Thank you very much for the introduction. You're saving me time. My name is Karen Saltoun. I'm a PhD candidate at Dr. Danilo Stokes Lab here at McGill University, affiliated with Mila AI Institute. And today I'm going to talk to you guys about brain asymmetry. I'm sure you know something about asymmetry, either from illustrations, on the web, or perhaps from the dictionary. The right side of the brain is often associated with creative and artistic thinking. The left side of the brain is more associated with logical reasoning. And these ideas are very prevalent, but they're also very simple. And I want to know whether such a simplistic viewpoint is really the best way to view the brain. Over the decades, functional asymmetry has been linked to both spoken and written language, handedness, sex, visuospatial attention, and more. Yet, despite how relatively well-established functional asymmetries are, even as recently as 2018, it's not clear which anatomical asymmetries arise at the population level, which is what I hope to address today. Looking into the literature, you will find researchers often focus on the cortical shell, taking pairs of homologous brain regions and asking are they larger on the left or the right and to which degree. Implicit in this is the idea that there's one archetypical pattern of asymmetry. Here, I don't mean a spatial pattern, but rather there's an archetypical collection of brain regions which co-vary with one another. And for simplicity, I'm going to refer to this as an asymmetry pattern. Today, I'm going to take a slightly different look at brain asymmetry and investigate the possible existence of multiple distinct brain asymmetry patterns. We can ask traditional questions to each of these asymmetry axes, and then an advantage of this approach is that it may naturally segregate different associated different associated phenotypes to different asymmetry patterns. In order to investigate this question, I'm going to capitalize on the UK Biobank, a large-scale repository of MRI scans for about 40,000 middle-aged individuals recruited across the UK. Part of the UKB Biobank protocol is a uniform MRI acquisition, processing, and expert curation, from which I identified pairs of homologous brain regions and constructed a targeted measure of both degree and direction of brain asymmetry. In this manner, I took 170 atlas parcels and computed 85 targeted measures of brain asymmetry, spanning cortical, subcortical, and cerebral volumes, and measures of white matter tract integrity. With this, I used a dedicated machine learning pipeline to identify common and cohesive patterns of brain asymmetry which pervade across the population. Using singular vector decomposition, we can identify one or many common motifs of brain asymmetry, and each will be associated with an importance score by construction, and that importance score is shown here. To orient ourselves, each point here is a single asymmetry pattern, with position on the y-axis indicating the amount of structural variation captured by a single asymmetry pattern. By construction, each of our asymmetry patterns is correlated from all other asymmetry patterns, and we have several asymmetry patterns that are able to explain more structural asymmetry variation than can be expected by chance, supporting the idea that there exist multiple distinct patterns of brain asymmetry. However, we also want to test the idea of whether these asymmetry patterns pervade across the whole brain. And in order to do that, let's zoom in on asymmetry pattern 1. Here we see the structural co-occurrence profile of asymmetry pattern 1. There's a lot going on, so let's go take it step by step. Each row here shows a different level of brain organization, and all are associated with asymmetry pattern 1, with darker colors indicating features with greater importance in describing asymmetry pattern 1. We see strong involvement of brain features across all examined levels of brain organization. For savvy individuals, you'll know that singular value decomposition will give us positive and negative loadings for each brain feature. By convention, I've chosen to plot positive loadings onto the right hemisphere, negative ones to the left, but there's also a sign invariance. So I'm going to go through what these loadings mean by focusing on the frontal pole. The frontal and temporal pole showed same signs, which indicate that volume asymmetries in the frontal pole co-occur with ipsilateral volume asymmetries in the temporal pole, i.e. they're larger in the same hemisphere within individuals. By contrast, the frontal and occipital poles had opposite signs, which indicate that asymmetries in the frontal pole co-occur with contralateral asymmetries in the occipital pole, and this pattern of contralateral frontal and occipital enlargement sounds an awful lot like brain torque, which is a phenomenon wherein the one hemisphere of the human brain is positioned slightly posterior to the other, causing a relative protrusion and enlargement of the right frontal and left occipital poles. This morphological characteristic is visible by eye, but has recently been shown to be absent in chimpanzees, thus potentially being human-specific. Importantly for us, we were able to recapitulate this well-known structural phenomenon in a purely data-driven way. Singular value decomposition knew nothing about this particular contralateral and frontal and occipital pole enlargement, but was able to organically highlight this pattern as the most explanatory pattern in our asymmetry. Beyond the brain, the UK Biobank is a rich repository of cognitive, physical, mental, cardiac health phenotypes, from which I extracted nearly 1,000 lifestyle and behavioral traits spanning 11 broad domains. I correlated each of these phenotypes to the individual expression levels of brain asymmetry pattern 1, and this is a result of our phenome-wide profiling analysis. To orient ourselves, each point shows the correlation, the association strength of the correlation between asymmetry pattern 1 and a single phenotype, with negative log 10 of the association strength being plotted on the y-axis, such that higher points indicating a stronger association strength between asymmetry pattern 1 expression and brain torque, or brain torque-like pattern. We see family size and physical, anthropometric, and blood assay levels are associated with asymmetry pattern 1. And for interest, I also highlighted right and left-hand dominance in the red and blue points here, from which we see that brain torque is significantly associated with asymmetry pattern 1. We also see a notable lack of associations with cognitive and mental health phenotypes, which is interesting because we know that brain asymmetry, functional brain asymmetry, is linked to to language. However, we also know that with a single model, we identified multiple brain asymmetry patterns. So let's zoom in on asymmetry pattern 2. Here is a structural co-occurrence profile of asymmetry pattern 2. Once again, we see involvement across all examined levels of brain organization, with particular emphasis on contralateral planum temporale and cerebral lobe 6 asymmetries. You may know the planum temporale as being associated with Wernicke's areas in the left hemisphere, which is an important part in the language comprehension profile and is one of the most prominent anatomical asymmetries found in humans. Here, we were able to organically highlight planum temporale asymmetries in conjunction with cerebral lobe asymmetries, and our phenome-wide profiling analysis showed particular emphasis strong associations with cognitive language phenotypes, as well as a plethora of mental health phenotypes and specific associations of socioeconomic strength. However, we also see that the asymmetry pattern 2 is not significantly associated with either right or left-hand dominance. In the interest of time, I've summarized our top 4 asymmetry patterns. Here, we see that the same brain region can be implicated in distinct asymmetry patterns. For us, this means that asymmetries in the planum temporale are separately associated with cerebral and hippocampal asymmetries. We also find that each asymmetry pattern has a unique phenotypic profile, which highlights differential behavioral associations to each of our asymmetry patterns. So overall, I hope I've convinced you that brain asymmetry can be uniquely thought of and captured with multiple uncorrelated brain asymmetry patterns, each of which has a unique phenotypic profile. We also were able to recapitulate well-known principles of brain asymmetry in a purely data-driven way, supporting the idea that these brain asymmetry patterns capture meaningful variations in structural asymmetry. I'd like to thank my lab and supervisor for all their support and encouragement throughout this work, and I look forward to answering any of your questions, either today or during my poster. And thank you all so much for listening.
Thank you very much. We have questions from the people. The total of variants explained by your model is rather low. Mm-hmm. How do you explain that more than 80% of the variants remain unexplained? Yeah, so our total explained variance is sums up to 100% by definition. So we know that our asymmetry pattern 1 is able to capture a pattern of asymmetry which is visible by eye. You know, looking at the brain, you can see that morphological characteristic. So to us, the relatively low explained variance of asymmetry pattern 1 doesn't mean that it doesn't capture something significant, but rather that there are multiple other characteristics of brain asymmetry that are as important or similarly important to brain torque. We found that 33 separate asymmetry patterns were able to explain more variation than could be expected by chance, which again may suggest that brain asymmetry is more complicated than previously thought, and may also contribute to why the literature thus far has been rather inconsistent if brain asymmetry in one region is associated with structural asymmetries across the whole brain separately. Very nice talk, thanks.
Oh, okay. Thank you very much.
And so our next speaker is Pedro Alves. So he will be looking at the subcortical anatomy of the ventral and dorsal attentional networks.
Thank you. So, as you know, attention is a core cognitive function that undertakes the filtering and selection of relevant information by the brain. It seems that attention is regulated by two main macroscale networks. The ventral attention network in orange is a stimulus-driven system, while the dorsal attention network mediates goal-driven processes. The study of their neuroanatomy has taken a predominantly corticocentric approach. However, there is evidence that these networks may also have subcortical components. First, subcortical electrophysiological recordings in primates has shown that subcortical structures such as the striatum and the pulvinar are activated during attention tasks. In addition, some distant phylogenetic species such as pigeons demonstrate some behavioral similarities with us during attention paradigms. Finally, in humans, neglect, which is a paradigmatic syndrome of hemispatial inattention, does not only arise after cortical lesions, but also after subcortical ones. We know that when we align different brains exclusively based on anatomical features, there might be a misalignment of small subcortical nuclei. In a previous work, we have demonstrated that if we do an additional step of functional alignment, we may improve the identification of these small subcortical structures. With this, the aim of our work was to explore the subcortical anatomy of the dorsal and ventral attention networks by the functional alignment of resting state fMRI maps. Our hypothesis was that some subcortical nuclei also integrate attention networks. To do that, we used 110 7 Tesla resting state fMRIs from the Human Connectome project in the MNI space. To compute ventral and dorsal attention network maps, we did a seeding analysis using Gordon's parcellation. This parcellation is composed by 23 cortical Venn parcels and 32 Dunn cortical parcels. For the functional alignment, we performed an iterative diffeomorphic transformation using ants, and the group effects were analyzed with one sample permutation test. For the structural connectivity, we used whole-brain deterministic stratigraphies. For functional connectivity, we calculated partial correlations. In addition, we also did a graph theory analysis to assess the hub properties of the identified nodes. Finally, we also wanted to assess the relationship between these new models of attention networks with the neurotransmitter systems. Specifically, we studied the spatial correlation between the projections of the identified subcortical nuclei with an atlas of 9 neurotransmitter systems that Justine Hansen has presented to us today. Here are our first results where you can see the surface projections of the ventral and dorsal attention network maps. As expected, these networks have different projection in the cerebral cortex and also at the level of the cerebellum. When we analyze the subcortical distributions of these networks, we can identify some of the hypothesized subcortical nuclei, such as the pulvinar, the caudate, and superior colliculi. In addition, we can see a group of other brainstem nuclei, such as the pedunculopontine nuclei and also the raphe pallidus nuclei. Actually, the conjunction analysis showed that most of these nuclei are shared by both networks. In terms of connectivity, the structural connectivity maps of the ventral attention network did not only show the association fibers, such as the third branch of the superior longitudinal fasciculus and the uncinate, but also projections fibers linking subcortical and cortical structures, namely the frontopulvinar and tetopulvinar tracts. The same happened with dorsal attention network, where we can see not only the first branch of the superior longitudinal fasciculus, but also frontopulvinar, parietopulvinar, and tetopulvinar tracts. In terms of the graph theory analysis, we measure two centrality parameters, namely between a centrality and degree centrality. And you can see that the subcortical nuclei have high ranks in both of these measures, suggesting that they do not only belong to these networks, but they seem to have or may have a position in the structural organization of these networks. Finally, when we analyze the special correlation between the projections of these brainstem nuclei with the distribution of neurotransmitter systems, we found three positive correlations, namely with the distribution of alpha-4, beta-2 nicotinic, acetylcholine nicotinic receptors, and with dopamine and serotonin transporters. With this, we propose an improved model of the ventral and dorsal attention networks that include the pulvinar, the superior colliculi, the head of caudate, and a group of other brainstem nuclei whose projections are specially correlated with the distribution of acetylcholine alpha-4, beta-2 nicotinic receptors, and dopamine and serotonin transporters. As limitation, our work is based on resting state analysis, so we are unable to untangle the dynamic interactions between the ventral and dorsal attention networks that occur during human behavior. However, future investigations with task-related fMRI in humans or electrical recordings in animals using our model may help to answer this question. Importantly, we believe that our model is consistent with the available psychopharmacological data, because we know that alpha-4, beta-2 nicotine agonists, methylphenidate and modafinil, which are inhibitors of dopamine reuptake and also selective serotonin reuptake inhibitors, have very well-known roles in terms of the modulation of attention. We also believe that our model reconciles behavioral data from distant phylogenetic species and ultimately may help to, or may contribute to a better understanding of attentional disorders in humans. Thank you very much.
Thank you. We have a question. How much smoothing did you use, and how does that play into the interpretation of the results?
Sorry.
How much smoothing did you use, and how does that play into the interpretation of the results?
I'm not sure if I'm understanding correctly the question. So,
how much smoothing did you use?
how much smoothing? Sorry, I was not getting. So we basically used like the Human Connectome project protocols. We didn't do additional smoothing to analyze the data. So we just did this additional step of functional alignment that does diffeomorphic transformation of the functional maps. So, and basically this does not change the gross anatomy of the brain. It just makes slight alignments or distortions of the functional map. So we didn't do additional smoothing. We just used like Human Connectome project protocols.
Thank you very much.
Thank you.
So our next speaker is Boton Dantal, with a headway in translation and research looking at brain age.
morning, everyone. So my talk is about a metabolic intervention to slow down brain aging. And I'll start with jumping right into the mechanism, just to make it clear the, apologies, to be clear about the mechanism that we are targeting with our treatment. So it's well established that brain aging involves hypometabolism, and glucose hypometabolism to be specific, and this hypometabolism is associated with dysfunction, atrophy, and the common clinical outcomes of brain aging. And if you go further up in this causal chain, there is evidence suggesting that there's actually insulin resistance. So brain aging and this glucose hypometabolism stems from insulin resistance. We actually hypothesized that what's happening is that glucose uptake gets impaired in neurons, in those neurons that use GLUT4 for glucose uptake. And this would imply that insulin resistance accelerates brain aging, and it's actually consistent with a meta-analysis that we published recently. So if you're interested in this field, I highly recommend you check it out. It's a pretty fast read. Another thing that this mechanism would imply is that if we use ketones, we can refeed the starving neurons. And the way that that works is ketones are alternative fuel source for neurons. So they bypass, I was just trying to refer to that middle figure. So as you can see, ketones use a different transfer mechanism. It does not involve, doesn't rely on insulin. So this way we can refeed the neurons. From a practical standpoint, ketones can be provided exogenously as a dietary supplement, but it can also be produced endogenously if you induce it via the ketogenic diet. So the question is, can we use ketones to reverse the aging-related effects? And in order to evaluate that, we dedicated a large-scale effort combining experiments on multiple spatial scales spanning from the cellular neuronal scale all the way up to the whole brain scale. And we also developed computational modeling tools to connect, to establish synergy among these experimental results in a mechanistic way. So today I'll be focusing on the experimental results on the two ends of this scheme, and I will start with the results on the neuronal scale. So we conducted experiments in mouse brain slices. We evoked insulin resistance, and we looked at what this would do to the speed of signal transmission in between brain regions. In this case, it was two subregions of the hippocampus. So when we added indinavir to the solution, which is an acute GLUT4 antagonist, so it blocks GLUT4, and this is how we evoke insulin resistance, we saw a sharp decrease in conduction velocity, the speed. It was about 15%, which is pretty significant in magnitude as well. And interestingly, once we added ketones, even at a low dose, at 0.1 millimolar, we saw these effects being reversed to the control level. And more interestingly, if we added in ketones at the physiological concentration, at 1 millimolar, we saw that conduction velocity actually increased above the control level, which might suggest that ketones could be more efficient, actually, compared to glucose. So now I'm going to move on to the whole brain scale. In this case, instead of insulin resistance, we looked at age as the main factor of interest. So we established a biomarker for brain aging, which also was a metric that we can modulate on short time scales, so that way we can eliminate confounders. We noticed that brain networks destabilized across the lifespan. We quantify this using brain network instability, which is essentially a dynamic functional connectivity metric with non-overlapping time windows. And at the end, we compress it down into a scalar using the R2 norm. So when we look at the age trends, we saw consistently across several publicly available data sets involving hundreds of subjects that networks destabilized across the lifespan. And what's interesting is it wasn't a linear increase, but if you look at those sigmoidal curves, you see that there's actually a sudden deterioration that starts at the age of 45, 50. So the question is, actually one more thought here. The fact that we saw this effect with EEG would suggest that it has to have, at least to a partial extent, a neural component as well. So it's not just a vascular effect or something on those lines. So next we ask, we come back to our ketones and we ask, can we reverse these effects? So in order to do this, we conducted a 7 Tesla fMRI study on human subjects. We administered exogenous ketones in these subjects, and we scanned their brain function right before and right after administering the drink. And we also repeated this paradigm with calorically matched glucose to control for the calories. We did this in 79 individuals. As you can see, the age range, it covers the adult lifespan pretty much up to the age of 80. And when we look at the results, what we saw is that unlike glucose, ketones were significantly stabilizing the networks, which is the opposite what we saw across age. So what this data set also allowed us to do is to evaluate the distribution of these stabilizing effects across the lifespan. But before I get into the results, I just wanted to spend just a minute on a concept that's relevant here. So in a lot of cases in biological systems, what we see is if we apply a strain on the system, we stress the system, initially we won't see any changes if the system will not dislocate from its baseline, because there is a feedback mechanism, a homeostasis that's keeping it in that point. But if we start increasing the strain, eventually this homeostasis will give up and we will suddenly dislocate the system and we'll start seeing changes. And this is the point, the interval where we should be seeing effects with an intervention, because this is where we can still reverse the seen effects. Because if we go even further, entering that red age territory with the strain, we actually start breaking down the system, and it becomes irreversible. There's not going to be any cells left to fix, essentially, simply put. So strikingly, if we go back to our results with the age trends, it is pretty much what we saw with our intervention. So as you can see, the age groups on the x-axis, if we start with the younger age group, we barely saw any stabilizing effects, which is consistent with the homeostatic state. But as we progress across age, we noticed the maximum extent of the stabilizing effects. And then once we get to the older age groups, the stabilizing effects started to diminish, where it might be beyond the breaking point. So to sum it up, these results suggest that metabolic intervention works, but we have to start early as the 40s. And above a certain age, it might not be effective anymore. And with that, I'll wrap up this talk. Thank you so much for sitting through this talk. And I just want to mention that our lab is hiring postdocs. So if you're interested or you just want to chat with us, stop by at our poster or reach out to us through our website, elsinore.org. Thank you.
Thank you. We have a few questions. Regarding the biomarkers, which exact networks were you referring to?
Could you repeat the beginning of the question?
Regarding the biomarkers, you were talking about networks, but which were those networks exactly?
I believe by biomarker I was referring to brain network instability, this dynamic functional connectivity metric.
Okay.
Hope that clarifies.
Then what kind of difference do you think you might see in your results among diabetic populations?
Diabetic population, very excellent question. That's exactly what we are up to. I mean, what we would expect is we would see the same effects, but it will be shifted to an earlier age range. So remember that I mentioned all the way at the very beginning that insulin resistance accelerates brain aging. So it's essentially the same process, it just happens earlier.
Thanks. So what about, sorry, I'm just reading the question. So what about the effect of episodic fasting, inducing a mildly ketotic state?
Yeah, excellent point. Yeah, exactly. So I forgot to mention that. The subjects are coming in a fasted state, which could be a mild ketosis. We collect blood ketone levels during these visits, and usually the ketone levels are very minimal, 0.1 to 0.2 millimolar. So I would say, at least based on the peripheries where we collect the blood samples from the finger, subjects are not in a ketogenic state. It's rather about being in a standardized state, so it would be unfortunate if someone came in after a big breakfast and someone else comes in after fasting for a day. So it's probably, that's why it's useful, not so much a ketogenic state.
And one last question. What was the ketone effect on an individual, sorry, I gotta rephrase that. Was the ketone effect at the individual level or at the group level?
What's the ketone? I'm sorry.
Was it like measured at the individual level or at the group level?
The experimental results?
Yeah.
Well, I mean, the conclusions would be on a population level, but as you can see, the design, it's actually within subject design, so we did all 4 scans on the same individual, so all the statistics are done in a within subject fashion, so it's individualized in that case.
Thank you.
Thank you so much.
Right, next we will hear from Sean Warrington about well, diversity and evolution in the brain.
Good morning, everyone. Thank you for the introduction. So evolution and development and its effects on the brain and organization occur at vastly different timescales, yet they've been shown to be linked. For instance, allometric changes show that there is overlap in which regions of the brain develop later in human development and those that have developed or evolved since common ancestry with primates and humans. Despite this, our understanding of brain ontogeny and phylogeny is still in its infancy, and we have a lot to learn. So measures such as allometry, although they've taught us quite a bit about this, are quite limited. And so we look towards structural connections to explore brain ontogeny and phylogeny instead. And these structural connections are closely related to area specialization in the brain, as structure defines functional boundaries. So we look to use structural connections to build a common space in which we can explore brain ontogeny and phylogeny. But in order to use these structural connections to do this, we need some common terminology across diverse brains. In other words, we need some landmarks. So our aim is to build this common space for concurrently mapping brain ontogeny and phylogeny. And towards this, we look to define consistent tractography protocols to construct landmarks. Now, these landmarks, we will use white matter tracts that exist across these diverse brains and are functionally equivalent, but rather than using their geometrical definitions, as they will be quite geometrically different across these brains, we use functional definitions. So, for example, the optic radiation, we know this connects the LGN of the thalamus to the occipital cortex, and it does not traverse hemispheres. So we define protocols to reconstruct this tract accordingly in the neonate, the adult, and the macaque. And we do this for 42 major white matter tracts for major association, commissural, limbic, and projection fibers in the neonate, the adult, and the macaque in respective template spaces. And we use our previously published tractography pipeline, FSL's extract, to perform tractography. So we do that, and we build these group average white matter tracts. And from these, we can see the overall homology of these white matter tracts across these brains, and in particular, the overall shape in the white matter bundles. But it's important to point out that these precise shapes and the precise endpoints of these tracts will differ across these brains, and it's these differences that we can use to explore brain ontogeny and phylogeny. So we now have these common landmarks, or white matter tracts, and now we want to encode their connectivity information at the cortex so that we can compare it across brains. And so one way we can do this is to basically find where these tracts cross the white-gray matter boundary and enter the cortex. And we could do this for the adult human, the neonate, and the macaque, and build a gray matter by tracts connectivity matrix for each of these. And since our tracts are equivalently defined and functionally equivalent across these brains, and they are equal in each of these matrices, we can compare rows of these tracts across ontogeny and phylogeny. For example, we could find rows of these matrices that have strong projections to occipital tracts, and we could contrast the connectivity in each of the brains. So far, my sort of description of how we build these matrices has been quite hand-wavy, saying that we want to find this intersection, but there's a problem, a limitation in tractography that we need to address before doing so, and that is the gyro bias. Many of us will be familiar in that in tractography, but there are two ways that we can, one way that we can overcome this, and that's by using a particular type of whole brain tractogram. And so there are two ways we can build whole brain tractograms. The first one is to seed in the white matter, but we have issues here. The fibers in the gyral area tend to bottleneck before fanning out towards the gyral walls. But in tractography, our streamlines struggle to follow this pattern and instead tend to bias towards the gyral crown. Instead, we can seed in the white-gray matter boundary, and now our streamlines can commence from this fanned out region and merge into the main fiber bundles, and this alleviates our gyro bias. So we can build a whole brain tractogram and then find the intersection between that tractogram at the cortex and our major white matter tracts from before to give us our white matter tract endpoints in the cortex. More specifically, how do we do that? So we build our whole brain tractogram, seeding from the white-gray matter boundary and targeting the whole brain white matter. And we count the number of streamlines or visitations to each white matter voxel, building a gray matter by white matter connectivity matrix. We then vectorize and stack our white matter tracts from before to give us a white matter by tract connectivity matrix. And now we have two matrices between which we can find the intersection through their multiplication, since they share the white matter dimension. And that gives us our gray matter by tract connectivity matrix, which is our common space. And columns of this common space represent maps of the tract termination on the cortical surface. Rows represent our connectivity patterns for a given cortical location. And we can repeat this for each of our three brains. And then, since our rows are consistently defined with these white matter landmarks, we can compare these rows statistically using a measure such as symmetric Kullback-Leibler divergence, for example. And this is a measure of distance between probability distributions. So for example, we could take a vertex in the occipital cortex of the neonate, and we could use KL divergence to sweep through the adult brain until we find the best matching vertex, and that would be minimizing our KL divergence. And we could repeat this for the macaque brain. And we would find three vertices with strong projections to the occipital tracts, for example. So we use this method, this approach, to build maps of the minimum KL divergence. And here, this is across ontogeny and phylogeny, comparing the macaque neonate, the neonate adult, and the macaque and adult brains. And we find that red regions here represent a greater minimum KL divergence. And this would indicate that in our common space, the closest connectivity patterns are further away. Overall, we find greater divergence across species than within species, as you'd expect, and some spatial variance in these divergence maps. Broadly, there is lower divergence in the sensory regions, such as the somatosensory area, and higher divergence in the association areas. Comparing across development, we see quite strong divergence in the inferior frontal cortex, and lower level divergence in the temporal area. Across species, we also see quite strong divergence in the inferior frontal cortex, but also in the temporal and parietal regions. We can use these maps to build what we term the joint divergence map. And this is simply multiplying the neonate human and the macaque human divergence maps from the previous slide. And here we get a map which represents how the neonate and the macaque diverge from the adult as a reference. So red indicates that they both diverge, i.e. neonatal brain regions that are developing later in human development, and also those that, regions in the human that have diverged from the macaque since common ancestry. And we see that this map overlaps and shares common features with previous maps of evo-devo cortical expansion. And then we can start to use this map to look at how connectivity patterns in different brain regions differ. And we see an interesting case in the inferior frontal cortex in Broca's area. And in BA45, we find a joint divergence. And BA45 is associated with semantic and lexical processing of language. So we see that the neonate and the macaque, the connectivity pattern diverges from the adult. Whereas in the BA44, which is associated with motoric and phonological processing of language, we see a phylogeny-specific divergence, whereas the neonate connectivity pattern is more similar. And in particular, we see that these divergence patterns are driven by the arcuate fasciculus, a language we know to be a tract we know to be a language tract. As well as these ontogeny-phylogeny divergence maps and these explorations, we can also target our framework towards exploring neonatal development more closely. So here, we compare different groups of neonates at different stages of development to the adult as a reference brain. We compare parcel-wise divergence across age, and we see that in the higher order association areas, there is more rapid development at this stage of the neonate development, whereas the lower order sensory areas are more stable. We can also use this framework to start looking at translating cortical atlases across brains. So here we take the similarity in connection patterns between the neonate and the adult, and the macaque and the adult, and we use spectral embedding to project these to a low-dimensional space. And we start to find that parcels from similar brain regions, such as the cingulate, cluster closer together in this low-dimensional space. And indeed, when we coarsely parcellate these this parcellation into lobes and take the center of gravity for each lobe, we find that they do, in fact, cluster together. And in particular, we see with the blue occipital cortex, these are clustered in the neonate and the adult, neonate adult and the macaque adult comparisons. Whereas in the frontal green, the frontal lobe, we see greater divergence in the macaque adult comparison, and this reflects what we know about the expansion of the frontal lobe in the adult and the human compared to the macaque. So, in summary, presented a unified framework for mapping brain differences in connection patterns across diverse brains. So this is a common space which is anchored on white matter landmarks, and these landmarks can be relatively robustly delineated using tractography. And we provide this protocol of tracts publicly, and these show correspondence across age and species. So finally, I'd like to thank all of the co-authors and collaborators on this, and our funders. If you'd like to talk more, then either send me a message or come to the posters later, or you can read our preprint. Thank you.
So we have time for one question. What do you do with tracts that do not have an equivalent between species?
Yeah, so this would be sort of a limitation in which tracts you can include in this framework. So in our case, we have 42 tracts, which we know are equivalent across these brains. Of course, if we move towards different species or different tracts that aren't equivalent, then perhaps they can't be included in this framework, and you need to seek some other representation that does exist.
But that does require postulate that they are like equivalent, really.
Yes.
Thank you very much.
Thank you.
So our last speaker for the session is Mei-Chi Niu. Mei-Chi works at the Research Center Jülich, and she will talk about receptor architecture and organization of macaque monkey, primary and secondary somatosensory cortex.
Thanks for your nice introduction. Hi there. I'm so glad to have this opportunity to share my work with you. Today I'm going to talk about the receptor architectonic organization of macaque monkey primary and secondary somatosensory cortex. The somatosensory cortex is involved in the processing of sensory information from across the body, such as touch, temperature, and pain. The monkey's primary somatosensory cortex is located on the posterior wall of the central sulcus and encroaches onto the free surface of the parietal lobe. Over the last few decades, research on monkeys has defined 4 strip-like areas within this region, namely areas 3A, 3B, 1, and 2. And each of these areas contains a separate body surface representation, which is arranged from the foot and leg on the medial cortex to the face and tongue on the lateral cortex. And the secondary somatosensory cortex is found on the lateral parietal and is almost completely hidden in the lateral fascia. The monkey's secondary somatosensory cortex consists of at least 2 areas. They are area S2 and the parietal ventral area PV. An understanding of how the somatosensory cortex processes sensory information depends on large part on knowing how it is organized. A complete and detailed parcellation scheme of the corresponding brain region can offer improved understanding of the biological substrate of brain functions, dysfunctions, and behavior. Therefore, the aim of the present study is to provide a defined architectonic map of macaque monkey primary and secondary somatosensory cortex. We not only wanted to analyze the cytoarchitecture of this region, but also want to obtain more information which could help us understand the combined structure and functional organization of the brain. So, the idea came up to use neurotransmitter receptors. Since receptors are key molecular which enable signal transmission between different nerve cells, and they are heterogeneously distributed throughout the brain. More importantly, the function of a brain region depends on a well-tuned balance between different receptor types. So, by visualizing the multiple receptor distribution pattern throughout the brain, we can not only identify the location and extent of the brain region, but can also get glimpse of their functionally relevant organization by assessing the degree of similarity of their multiple receptor balance in each identified area. So let's take a look at the method we used. We analyzed the brains of 3 adult macaque fasciculars monkeys. The brains were removed during autopsy. Each hemisphere was deep frozen and sectioned into chronal sections of 20 micrometer thickness. Alternating sections were incubated with tritiated ligands or stained for cell bodies. As an example, here I show a group of sections from the same roster caudal level. For this group of sections, we can obtain information of 15 different receptor types and the corresponding cyto and the myelo architecture. That means in this very tiny brain region, we have more than 15 modalities to study. After digitization and color coding, we can obtain clear visual impression of the laminar and regional distribution pattern of different receptor types, which were used to identify the borders in potential borders in between different regions. So here, an observer-independent and quantitative statistically testable method was applied for detection of the potential border in both cyto and receptor architecture. The potential border was accepted only after confirmation of its statistical significance. Moreover, the biological relevance of the potential border was confirmed by comparing spatially corresponding borders from the adjoining sections. At the end, for each identified areas, we extracted the densities of all analyzed receptor types for the further statistical analysis. In the present study, 13 architectonically distinct areas were identified for better understanding of the topographical relationship towards neighboring areas. Here I show a series of chronal sections superimposed with corresponding areas. Within the primary somatosensory cortex, we confirmed the existence of the previously described areas 3A, 3B, 1, and 2. Additionally, we identified medial and lateral subdivisions of 3A and 3B respectively. Furthermore, the latter part of 3B could be subdivided into an internal and an external subdivisions. Within secondary somatosensory cortex, 6 identified areas are arranged in 3 roster caudal zones. So here I show an example of the receptor architectonically defined borders between 2 3BL subdivisions. This colorful picture depicts the GABA-A receptor distribution pattern in a coronal section through macaque monkey central sulcus, and red codes for high, loads for low receptor densities. The borders between 3BLI and 3BLE are reviewed by changes in GABA-A receptor densities. All other examined receptors also review this border, either by overall density differences or by changes in specific layers. It's worth noting that not all borders are equally clearly defined by all receptor types. Later, the absolute mean area density of all analyzed receptor types were quantified and projected into the corresponding area. That means, in addition to provide a redefined architectonical pastillation scheme, in the present study, we further provide 15 different receptor distribution maps for macaque monkey primary and secondary somatosensory cortex. And one of the main purposes of our study was to analyze the monkey somatosensory cortex with a focus on the relationship between its molecular and functional segregation. So for each identified areas, the 15 receptor density were visualized as receptor fingerprints. So that means each area has its own characteristic receptor expression pattern. And the similarities of their receptor fingerprints enable the segregation of these areas into 3 functionally relevant groups. First, multivariate analysis reviewed a clear segregation between primary and secondary somatosensory areas. Furthermore, within primary somatosensory cluster, area 3AL and all subdivisions of 3B are found in one cluster, and 3AM, 1 and 2 in the other one. So here we have termed the former cluster as S1 proper cluster, since areas 3B, 1 and 2 form a hierarchical sequence of processing, and area 3B is thought to constitute a true primary somatosensory cortex, which has chemoarchitectonic features that are characteristic as a primary sensory area. Well, areas 1 and 2 had less dominant sensory features, which resemble those of secondary sensory areas. This notion is further supported by the segregation between all 3B subdivisions from areas 1 and 2. So now I would like to summarize my talk. First, we reevaluated the pastillation scheme of macaque monkey primary and secondary somatosensory cortex, and we created this 3D map which has been brought into stereotaxic space. Second, we created 15 receptor distribution maps for this region. All these maps will be publicly available via the Human Brain Project and the BASA repositories afterwards. At the end, we provide insights to clarify the functional organizational model of this region. Last but not least, I would like to thank my colleagues for their support, especially Professor Nicola Palomero-Gallagher, she's here, and also Professor Katzelas. And thanks for your attention. If you would like to hear more about my talk, I'll be very happy to welcome you at my poster. Thank you all.
Thank you very much. So the most popular question was, is there any differences in the structure and organization of the sensory cortices between human and macaques?
Yeah, that's a very interesting question. And we, actually, I have no chance to check whole somatosensory cortex for human. But for especially the true primary somatosensory cortex for human and monkey, that's all 3B, and they have very similar receptor distribution patterns. For example, they have really extremely high M2 receptors when we compare 3B with other somatosensory areas. Yeah, that's it.
Thank you very much.
Thank you all.
Apologies. So this concludes our session. Thank you everyone for attending.